Welcome back to the deep dive. Today, we are diving deep into, well, one of the most diverse and really influential groups on the planet, the gram-positive bacteria. If your idea of these microbes is just sort of simple, uniform rods and spheres, maybe prepare for a bit of a shock. We've pulled key sources from Prescott's Microbiology, and we're going to show you how this phylum is responsible for basically everything from the smell of fresh earth to like two-thirds of the antibiotics you've ever been prescribed.
Yeah, absolutely. It's an immense and really crucial area of microbial study. Our mission today is to move beyond just that simple stain test and navigate the complexity of their structure, their classification, specifically thinking about the high G plus C versus the low G plus C groups, and, extract those crucial details about their function and their huge relevance in health and industry.
And we're kicking off with a story that, honestly, might fundamentally change how you think about antibiotics. We've always seen them as, like, biochemical weapons, right? Microbial warfare. But the source material suggests they might be something more fundamental, maybe a sophisticated form of bacterial communication.
Exactly. When we talk antibiotic production, the genus Streptomyces is, well, the undisputed champion. These are soil bacteria, Actinobacteria actually, and they're responsible for producing an estimated two-thirds of all prescribed antibiotics. We're talking familiar names like neomycin and tetracycline.
Two-thirds, that's incredible. And the arsenal doesn't stop there, does it? The sources talk about anti-cancer drugs like doxorubicin, antifungals, even antihelminthics like ivermectin.
It's a massive chemical factory. The
Oh,
sheer scale of their potential is just staggering. While they produce maybe 10,000 known bioactive products, scientists estimate there could be closer to 1 million more undiscovered compounds just locked away in their genomes.
whoa, 1 million. So why haven't we found them?
Well, what's fascinating here is the sophisticated regulatory system that kind of keeps them hidden. When researchers sequenced S. color, a strain known to make 4 antibiotics, they actually found the genes for 20 more. But these genetic pathways are tightly regulated, basically governed by a molecular off switch.
An off switch. Huh. Why would an organism, have the genetic blueprint for its own weapons, which must cost energy to make, and then wait for some kind of signal to actually deploy them?
It seems to be a matter of evolutionary hedging. Why waste energy on defense unless the neighborhood is getting crowded, right? That lock is only opened by specific chemical signals from other microbes. For instance, streptomycin production needs the release of A factor, a small molecule that acts as an intercellular signal. Other compounds like AHFCAs control other complex metabolite releases.
So when we culture these streptomyces isolates alone in a nice, clean lab.
They often stay silent. They need that communication, that signal that the environment is challenging to unlock their full chemical repertoire. And this has, well, massive implications for drug discovery. It really suggests we need entirely new culturing methods that mimic their complex social life in the soil.
Okay, that sets the stage perfectly for the enormous task of classifying this group. We need a systematic way to manage all this diversity. And for gram-positives, the first big division is based on their DNA, right? The concentration of guanine and cytosine.
Correct. We use the G plus C content of the genome. We have the high G plus C group, meaning above 50 mol% G plus C. That's dominated by the phylum Actinobacteria. Then we have the low G plus C group, the phylum Firmicutes, which is home to the classes Bacilli, Clostridia, and the more recently defined Negativicutes.
Right. Let's start with the Actinobacteria then, our filamentous high G plus C group. These are the microbes that produce geosmin, right? The compound responsible for that characteristic fresh, earthy smell after rain.
That's them. They are chemorganoheterotrophs, and they have a complex multicellular life cycle, way beyond just a simple single rod. They form these long, branching multinucleoid cells called hyphae. Think like fungal filaments. These hyphae develop into a network, or mycelium. There's the substrate mycelium, which grows down into the medium or soil, gives colonies that dense, leathery texture.
And then the aerial mycelium grows upward, and that's often linked to making those famous secondary metabolites, the antibiotics.
Often, yes. It signals a different stage in their life cycle.
So how do these soil residents survive drying out, especially up in the topsoil?
Through spore formation. But it's really crucial to distinguish their spores from those of, say, the Firmicutes. Actinobacteria produce exospores. These are formed when the aerial hyphae basically segment and pinch off. They don't develop inside a mother cell like endospores do.
Ah, okay. So not the super heat-resistant endospores we hear about.
No, not like those. But they are highly effective at withstanding desiccation, drying out, perfectly suited for that topsoil environment they live in.
Taxonomy here sounds a bit tricky. You mentioned the sources say 16S RNA sequences aren't always enough to tell genera apart. So what other microbial fingerprint do scientists rely on?
We have to look really closely at cell wall structure. The peptidoglycan structure across Actinobacteria is incredibly diverse. So microbiologists analyze the specific amino acids used in the interpeptide bridge, the bits linking the glycan chains. A key example is diaminopamelic acid, or DAP. Identifying these unique peptidoglycan components gives us that molecular fingerprint needed for classification when, other methods aren't quite specific enough.
Okay, let's move to some signature genera within this group. We mentioned Corynebacterium, known for its distinctive snapping division. What exactly causes that palisade fence post look?
Right, the palisade arrangement. It's a neat visual illustration of mechanical tension, actually.
Mm.
Corynebacterium has a two-layered cell wall. When the inner layer grows inward to form the septum for division, the outer wall layer is pretty rigid. It doesn't stretch evenly. Tension builds up until the outer layer just abruptly ruptures at its weakest point. Exactly. But because a small piece often remains attached like a hinge, the two daughter cells are left sort of resting at an angle to each other. That gives us that distinctive palisade arrangement, like a row of fence posts. It's a unique structural result of how they divide.
Fascinating. And its famous pathogen, C. diphtheria, causes diphtheria. Okay, that leads us to perhaps the most structurally complex gram-positive, Mycobacterium. Why does it take them so long, like up to 40 days, to grow in a lab?
It's almost entirely down to their specialized cell wall. It's highly hydrophobic waxy, which makes nutrient uptake incredibly slow. While they do stain gram-positive, technically, their peptidoglycan is contained within a sort of periplasm, and it's covered by an additional really thick outer barrier made primarily of these complex lipids called mycolic acids.
So it's not like the LPS outer membrane we see in gram-negatives, but more like a thick, waxy shield of fat.
Precisely. These massive mycolic acids are covalently linked to the peptidoglycan layer via a polysaccharide called arabinogalactan. This lipid shield is what makes the cell wall so impenetrable to most organic molecules, which is why they show such natural resistance to common antibiotics. It's also the chemical reason why they're acid-fast. They resist decolorization by acid alcohol and staining, which is a critical diagnostic technique.
And these are the agents of ancient diseases like tuberculosis, M. tuberculosis, and leprosy, M. leprae. Heavy hitters. Moving on, we have the nocardioforms like Nocardia and Rhodococcus. What's their big role in the global context?
Their real importance lies in environmental cleanup, what we call bioremediation. They have this incredible metabolic versatility that lets them degrade really complex, tough molecules. Things like petroleum hydrocarbons, detergents, even highly toxic pollutants like polychlorinated biphenyls or PCBs. They're basically the ultimate environmental decomposition crew, though Nocardia asteroids can be an opportunistic pathogen in humans too.
And finally, within Actinobacteria, the sources mention Arthrobacter, which shows an unusual survival mechanism called the rod-coccus growth cycle.
Yeah, this is a really beautiful example of microbial adaptation to changing conditions. In times when nutrients are abundant, that's the exponential growth phase, they exist as active, often branched rods. But when the environment gets harsh, maybe dry or nutrient-starved, they shift into a non-spore-forming but still resistant cocoid form, little spheres. This allows them to survive the stationary phase much better until conditions improves. Then they can switch back to rods.
Okay, so if the high G plus C group represents these sort of slow, deliberate chemists of the soil, the low G plus C group, the Firmicutes, is maybe the one we're most familiar with in terms of, well, pathology and food science. Let's dive into the class Bacilli now.
Right, and the genus Bacillus is absolutely central here.
Huh.
Bacillus is a crucial model organism. It's studied globally for everything from complex gene regulation to the exact mechanism of endospore formation, and even multicellular behaviors like forming intricate biofilms and structures that look a bit like fruiting bodies.
And their practical applications are huge, right? Producing antibiotics like bacitracin and polymyxin. But the use that always catches my eye is how they're weaponized as a natural insecticide.
Yes, that's B. thuringiensis, often called Bacillus. During the process of forming its endospore, this bacterium simultaneously produces a massive, solid protein crystal right next to it, called the perisporal body. This crystal contains toxins, delta endotoxins, that are completely harmless to us, in mammals. But when ingested by a susceptible insect larva like a caterpillar, the alkaline conditions, the high pH of the insect's gut, solubilize the crystal and activate the toxins. These toxins then basically poke holes in the gut lining, destroying it.
Wait, hold on. So this tiny organism literally packs a ready-to-use crystalline bomb that only detonates under very specific pH conditions inside its target. That's incredible evolutionary engineering.
It really is highly specific and effective. Of course, the darker side of Bacillus is B. anthracis, the bacterium that causes anthrax.
Right.
Now, shifting from rods to cocci within Bacilli, we have Staphylococcus, famous for those irregular grape-like clusters you see under the microscope. We really have to focus on S. aureus, the master pathogen. It seems like it's evolved every trick in the book to cause disease.
It is definitely armed to the teeth. The sources highlight its really staggering collection of virulence factors.
What's maybe the cleverest trick it uses to dodge our immune system?
Ooh, good question. I would probably argue it's protein A. This is a surface protein that does something really counterintuitive. It binds to the efficacy region of our own antibodies, that's the handle part, that our immune cells, like phagocytes, usually recognize and grab onto. By binding the antibodies essentially backward, S. aureus effectively camouflages itself. It renders the host's primary antibody defense system useless for targeting it.
Wow, like putting a bag over the immune system's targeting scope.
Yeah, that's a pretty good analogy. It also uses tools like coagulase to form clots and hide initially, and then staphylokinase to dissolve those clots later, helping it spread and invade deeper tissues. That's a really sophisticated attack profile. Okay, now we move quite abruptly from aggressive pathology to delicious utility with the lactobacilli, the lactic acid bacteria, or LB. Their defining metabolic trait is what makes our yogurt tangy, right?
Yes, exactly. They are strictly fermentative organisms. Key point. They lack an electron transport chain and cytochromes. They rely entirely on substrate-level phosphorylation to make their ATP energy. Lactobacillus species typically perform homolactic fermentation, where lactic acid is virtually the only major end product. And that process is indispensable for making things like hard cheeses, yogurt, sauerkraut.
But not all fermentation is the same in this group. What's the difference when we look at leuconostoc?
Good point. Leuconostoc carries out heterolactic fermentation. They use a different pathway, the phosphochitolase pathway. This produces not just lactate, but also ethanol, or sometimes acetic acid, and carbon dioxide gas.
The CO2, the bubble.
Exactly. That CO2 production is crucial. It provides the characteristic holes in cheeses like Swiss, the effervescence in some drinks, and just adds texture and complexity to many fermented foods, like kefir or some vegetable fermentations.
Got it. And finally, under the Bacilli umbrella, we have Streptococcus, identified clinically by how they break down blood on blood agar plates, the hemolysis patterns.
Right, we look for how they lyse red blood cells. Alpha hemolysis gives you this kind of greenish zone of incomplete breakdown around the colony. That's the classic signature of organisms like S. pneumonia, major cause of pneumonia. Then there's beta hemolysis, which creates a completely clear zone of lysis where all the red blood cells are destroyed. The most infamous behemolytic pathogen is S. pyogenes, also known as Group A strep, responsible for strep throat, scarlet fever, and more serious invasive diseases.
Okay, now we shift gears again, moving to the class Clostridia. These are the obligately anaerobic endospore-formers. They thrive where oxygen is absent and are, well, responsible for some of the most potent neurotoxins known to science.
Yeah, because they absolutely must operate in anaerobic environments. Think deep soil, sediments, improperly canned foods, or even deep wounds. Their fermentative metabolism is highly distinct. Many Clostridia rely on something called the Stickland reaction, which is essentially a coupled amino acid fermentation.
So they use two amino acids together, not just sugar.
Exactly. One amino acid gets oxidized, which generates some ATP for the cell. Meanwhile, a different amino acid acts as the electron acceptor and gets reduced. This coupling allows them to get energy just from amino acids, and it generates ammonia, plus these volatile fatty acids, things like butyric acid, fuleric acid, often the foul-smelling compounds that are characteristic of putrefaction or protein decomposition.
Right, putrefaction. And we absolutely can't discuss Clostridia without mentioning the big pathogens, C. botulinum causing botulism and C. tetani causing tetanus. But the metabolism of C. tetani presents one of the most, well, surprising examples of microbial engineering in our source material. It can't even use sugars.
It truly is fascinating. C. tetani lacks the ability to catabolize sugars. It primarily uses that Stickland reaction. But here's the twist. Even though it's strictly fermentative, it maintains its ability to take up nutrients by using a specialized V-type ATPase enzyme to actively pump sodium ions, Na+, out of the cell. This doesn't create the standard proton motive force, PMF, that most bacteria use across their membrane. Instead, it establishes a sodium motive force, or SMF.
A sodium gradient instead of a proton gradient.
Exactly. And this SMF, this sodium gradient, is then immediately used by other transport systems to power the uptake of nutrients into the cell. There's even an unusual electron transport chain linked to NADH oxidation, but its purpose seems to be extruding sodium ions, not respiration with oxygen.
Wow, that is highly unusual, using a different ion force, sodium, and linking ATP hydrolysis and electron transport to sodium extrusion rather than respiration. It just shows the extreme lengths a strictly anaerobic organism has to go to just to acquire basic nutrients from its environment.
It really highlights the incredible metabolic ingenuity required to survive in oxygen-free environments, which unfortunately often comes with a pathological price when these organisms find their way into a host. Oh, and before we leave Clostridia, we absolutely have to note the phototrophic anomaly, the Heliobacterium.
Right, phototrophic, like using sunlight, but also anaerobic, endospore-forming, and classified within the Clostridia. That sounds like a microbial identity crisis.
They are truly unusual, yeah. They are anaerobic phototrophs, they do form endospores, and they use a unique pigment, bacteria chlorophyll G. But critically, they lack the complex internal photosynthetic membranes like thylakoids or chlorosomes that are common in other phototrophic bacteria. Their photosynthetic pigments are simply contained within the regular plasma membrane. It just shows that even within a class largely defined by strict anaerobism and fermentation, evolution can still find a path to harvesting sunlight energy.
Okay, let's wrap up this deep dive with the newest and arguably most revolutionary class mentioned, Negativicutes. This seems to force us to question the very definition of gram-positive altogether.
This is definitely where classification gets fundamentally revised, based on molecular data.
Mm.
Isolates within this class, like the type species Negativicutes caecina, are defined by their 16S rRNA gene sequence as being genetically related to the gram-positive Firmicutes. They belong in that low G plus C lineage. However, physically, when you look at their cell structure, they possess two distinct membranes, an inner cytoplasmic membrane and an outer membrane, what microbiologists call a diderm structure.
Like a gram-negative bacterium.
Exactly like a gram-negative bacterium in structure. And because of that outer membrane, they stain gram-negative in the standard gram stain procedure.
So they look gram-negative, they stain gram-negative, but genetically they're low G plus C gram-positives. What is the evolutionary implication of this discovery? This seems huge.
It is pretty significant. The prevailing theory now is that the common ancestor of the entire Firmicutes phylum might have actually been diderm possessing an outer membrane, and that this outer membrane was subsequently lost multiple times independently during evolution in most lineages.
Lost. Why would they lose it?
Well, one strong hypothesis is that losing the outer membrane might have been facilitated by, or even necessary for, the evolution of the complex physical process of endospore formation. Arguably, forming that incredibly complex multi-layered spore structure inside the mother cell is physically easier to accomplish with only a single membrane structure to manage, rather than two. The intense selective pressure for robust endospore formation might have literally shaped the cell envelope evolution of the entire phylum.
Wow. So the need to make spores might have driven the loss of the outer membrane in most Firmicutes. And practically speaking, members of this newly defined class, like Veillonella, are actually part of the normal human biota, right? Living in the mouth and the GI tract, which shows just how deeply this revised taxonomy impacts our understanding of even our own microbiome.
Absolutely. So we've covered a massive amount of ground. We've moved from the filamentous antibody-producing high G plus C actinobacteria through the powerful endospore-forming low G plus C Bacilli and Clostridia with all their metabolic tricks, and finally arrived at the Negativicutes, who kind of force us to acknowledge the fluidity of even the fundamental gram stain definition. It's just a remarkable illustration of how microbial structure dictates function, for those waxy mycolic acid barriers dictating slow growth and resistance, to using sodium motive forces, to maybe even shedding membranes for spore efficiency.
And let's maybe end where we began, thinking about the vast untapped potential of the Streptomyces. We know these soil chemists have the genes locked away to produce dozens, maybe hundreds, of potentially life-saving compounds, but they only seem to unlock them when they get the right signals from their microbial neighbors. As a learner, really recognizing that we might be missing out on, well, maybe a million potential drugs just because we aren't listening in on the bacteria's chit-chat properly, that should really give you pause.
It absolutely should, and it encourages a new kind of thinking in microbiology and drug discovery. Understanding the metabolic and structural complexity of these organisms, From the slow growth challenges of acid-fast bacteria to the evolutionary push that perhaps led to the negativicutes is really the first step toward finding the next generation of therapeutics, better environmental solutions, and even new food technologies. These tiny organisms drive major global processes.
A perfect place to leave it. Thank you so much for taking this deep dive with us today into the world of gram-positive bacteria.